Hello there, my name is Rob Durham.
And I'm John Chappell.
And you're listening to another episode of the Gyrolab Spin. Well, welcome back, John, and everybody else. It's today's topic. We're going to be talking about the generic ADA assay using the Gyrolab system.
Yeah, so this is particularly used for preclinical ADA assessment. So we're going to have a little bit of debate today about the utility of using ADA assays in preclinical and what assay formats to use. So that's going to be part of the discussions.
Yeah, I think this is an important topic for many of our customers, especially those working in that preclinical space. So it's going to be good to have this discussion.
So the outline of today's podcast is that we're going to give an introduction to ADA, anti-drug antibody. We've already done a podcast on ADA assessment, so this is really just a quick overview. Then we're going to go through an interesting survey that was done by Bioanalysis Zone and to kind of understand the market for generic assays and the utility of generic assays. And then we're going to give an introduction to the new Gyrolab product for assessing ADA in preclinical species using a generic format.
Yeah, no, this sounds really good. I'm excited to have the discussion.
Yeah, it should be good. So immunogenicity, or the measurement of anti-drug antibodies, is an important part of biological development, and it's pretty much used in both preclinical and clinical trials for large molecules. It's important because it can have important clinical implications, things such as allergic reactions, things like immune complex toxicity, which potentially can block things like your kidneys and other organs. It can actually cause the body to start reacting against itself, so causing autoimmunity, and also it can cause reduction of efficacy. And when you're developing immunogenicity assay, it's always a balance between drug tolerance and sensitivity and target interference, because most biological drugs have very, very long half-lives, so they're in your system for many days, even months. So obviously you're trying to measure an anti-drug antibody in the presence of drug. Also, if there's a soluble target, then you have to take that into consideration. So its sensitivity against the drug concentration is the fundamental part of ADA assessment.
And I would say that's one of the biggest challenges I've seen in the preclinical space is the notion that you've got to balance these challenges of drug tolerance and target interference to accurately measure the presence of an anti-drug antibody.
Yeah, I had a situation in a preclinical study when we needed to get drug tolerance for up to mg per mil of drug. Yeah. For most clinical studies, we're happy with drug tolerance around 100 micrograms per mil. So, in toxicity studies, you dose in much higher doses. So, but obviously, it's not as critical as clinical, for safety reasons. But it is important sometimes when you're understanding or interpreting the PK or any other safety issues in the animals.
If you have some of those safety signals in kidneys and so forth with some of these drugs in the preclinical space, they're going to want to understand those relative to the TK, the toxicokinetics of the drug.
Exactly. When it comes to the development and validation of immunogenicity assay and the approaches used, these have very much come from industry white papers. So the Myers-Luce and the Shankar papers have really defined how to validate these assays. And these have very much made it into the regulated guidance, both the EMEA and also the FDA. But what hasn't made it across actually is the guidance specifically for preclinical. So we're going to discuss that today.
Excellent.
So as mentioned, these white papers have very much driven the industry regulatory agencies to come up with their own guidance. And an example here is the FDA guidance, the guidance for industry. And what this actually has done is lowered the expectation for sensitivity requirements. So now in the old papers, it requested kind of 200 to 500 nanograms per mil, and that's been lowered to 100 nanograms per mil. And also this means that we need increased drug tolerances required, and as it is expected at the sensitivity level of the assay. So it means that for a lot of assays we need to do some sort of pre-treatment. such as acid dissociation, to actually ensure that we can measure the antibody in the presence of high level of drug. And there actually is no industry guidance for preclinical assessment. And what this has actually meant that is a lot of people are applying the same criteria for preclinical assays as they are clinical assays, because it's like a vacuum. So, but there isn't the same expectation when it comes to preclinical assays. And today, really, we're going to kind of discuss generic approaches, because we feel that these could be applicable for many preclinical studies.
Yeah, I think from my perspective, I've seen folks be overly conservative, maybe. One could argue that they do a lot more to develop preclinical ADA assays look very much like a validated clinical assay, when, you know, maybe that's not entirely appropriate, maybe it's a bit of overkill, and doesn't really recapitulate in the preclinical side what may happen in the clinical side anyway. And so given the lack of translatability, is that the best approach?
Yeah. I mean, sometimes if you've developed your assay in the preclinical space, it makes it easier when you move forward into your clinical assay, because you have the basis for the assay. And that's one of the reasons why people do use kind of the similar approach. But you know, sometimes like 40, 50 runs of validation is certainly not required for a preclinical assay, but some people are doing it.
Yeah,
so.
Exactly. So it'll be worth having a discussion about whether that's a better use of your time, especially when you're in the earlier phases of drug development, and not all of those candidates are going to move forward.
Yeah, and ultimately, a fully validated assay is only required for regulated submission for a clinical. So it's not necessarily required for a preclinical assessment. So.
Yeah, that's good.
So following on from the white papers and the regulatory guidance, pretty much everybody is following this tiered approach, particularly for clinical study, where you have a screening assay and you validate and develop a screening and confirmatory assay. You have a screening tier. If it's positive, then you confirm, normally by adding additional drug, and then you go for further characterization depending on the stage, things like a neutralizing assay. So this is kind of the standard approach. When it comes to preclinical, there's a lot of debate of what is needed. Certainly a lot of people are doing screening assays and maybe just doing titer, for example, or they're avoiding doing confirmatory at all. So there is no clear guidance on what to do in preclinical stage. There has been a few position papers on this, but obviously there isn't, certainly not a need to do the full characterization as you would in a clinical assay.
So what we'd like to do now, John, is sort of take a look at some survey results on the utility of generic ADA assays within the bioanalytical community. And so where this comes from is we had seen some of our customers develop sort of a generic approach on the Gyrolab system, and we'd had certainly some requests from other customers who were working in the preclinical space where you have lots of different drug candidates that are moving through the pipeline, and yet they still had the need to understand the potential for an anti-drug antibody response. And so what we wanted to do is marry those two ideas together and see if it made sense for us to come up with a sort of off-the-shelf solution and sort of get into this a little bit. We wanted to get this information from the community with help from the bioanalysis zone. Is that okay?
So the first question in the survey is, does your organization currently use generic ADA assays? And you can see here actually that approximately 40% are actually currently using generic assays, but the slight majority currently used and don't use generic assays or use drug-specific assays.
Yeah, I think that probably mirrors my experience. The majority of the people that we've worked with, I would say, are using a drug-specific assay in the preclinical space.
Yeah, I agree.
In this question, the question was, would your organization be more likely to implement a generic ADA assay during the biotherapeutic development if a kit were available? And you can see quite a large majority of people would. And so, I mean, this gives us confidence that maybe having a generic option that's off the shelf for our customers to use would be a good option.
Yeah, I think this really gave us confidence that a Gyrolab assay for a generic assay with ADA would be important.
Yeah. So this question is subtly different from the previous one. Would a generic ADA assay kit be useful to your organization if it were available? And I think, it looks like a majority of the respondents said that they would find that data from a generic assay useful in their preclinical development process. Of course, there are still some folks out there that would be resistant to that, I think, because again, for tradition or their own perceptions, they feel like having a drug-specific anti-drug antibody assay is the way they've been doing things, and so they'd be a little bit reticent about moving to a generic method.
I think you're right. A lot of people are very conservative, so they want to do things the way they've always done them. So even though specific assays can cause issues, particularly for drug tolerance, with high drug concentrations, it's quite hard to measure early onset ADA, for example.
Exactly. So, and then I think it can make interpreting PK results challenging, and then especially when you're working in that preclinical space, I guess maybe they implement them further down the development scheme when you're out where you're really getting ready to make that leap with the IND work into the clinic.
So in terms of which study types do people use anti-drug antibody assays, so you can see that many people use it in early phases for discovery and lead selection, but also in preclinical, particularly for things like dose ranging, finding studies, etc. But mainly immunogenicity is used in regulated work. But immunogenicity assays early in discovery can show you whether maybe you may select the least immunogenic molecule, although obviously immunogenicity in animals is not always predictive of immunogenicity in clinical, but you can see really that people are using immunogenicity right through drug discovery and drug development.
Yeah, and I think here maybe this is part of the issue that you run into is you're in regulated preclinical drug development, and so the idea that you're in regulated, that's why we need a validated ADA assay. And if they have the perception that it's going to short circuit their work in the clinic, then maybe that's part of the justification why you see that little bit of a reticence about using a generic assay for the immunogenicity work.
No, exactly.
So in this question, the answer was, if you said yes to the generic ADA assays, do you consider measurement of complexes to be an effective assay strategy? And here you can see a majority of folks would say that it was fairly highly effective. And I think that speaks to the fact that, well, if there's, since we're using the anti-human, you would use the anti-human as the capture reagent in this. It's then an anti-drug antibody would have to be complex to the drug in that case to be able to see a signal.
Yeah, and the advantage of this format is actually you're pulling down the complex, but you also can detect the immune complex a lot earlier because it's more drug tolerant, which we're going to go through in the next few slides.
Yep. So then, in summary of the survey results from the bioanalysis zone, what you can see is that the generic assays can be used to support drug development in the minds of the folks that took the survey, as well as the fact that a significant number of folks are already contemplating or using a generic format in their anti-drug antibody assays.
Yeah, and the availability of a commercial kit may increase the generic ADA use, and what that will do is support drug development and means that you have an off-the-shelf method that can be easily used without method development. So, as I said, there is no specific guidance that limits the use of generic assays, The guidance is out there generally for clinical, supporting clinical studies. So there's no reason why a generic assay can't be used. And there's certainly a move in the industry for less immunogenicity to be done automatically as part of preclinical, and mainly only when it's required or you get unexpected PK, etc.
Yeah, and so I mean that really leaves us with the last point here in the survey that this immune complex measurement is a viable alternative in the mind of a lot of folks to measuring the traditional free ADA that you do with labeled drugs. With that leaves us with the potential for an off-the-shelf solution, and so that's what we want to turn to next, is talk about what Gyrolab's been doing in this space to sort of come up with an off-the-shelf solution that folks could use to do generic ADA.
So now we move on to going through the generic Gyrolab ADA assay and how this particularly can be used to support you.
So the advantages of an ADA kit then are that it really streamlines your candidate selection. If you have at the stage of the drug development process or early discovery where you're looking at multiple drug candidates, you don't want to be developing an individual assay for each drug.
Yeah, and then it means you reduce the amount of assay development and optimization. You can have an off-the-shelf method that then can be used to screen all your drug candidates. And I think this is a major use for this product is at that stage.
Yeah, and then of course this facilitates the sort of ADA screening that happens in that non-regulated preclinical studies as you move the process forward, and then it really helps then put in context the PK findings that you might see that don't make sense. When you see an aberrant PK finding, then the presence of this circulating immune complex could indicate the rationale for why that PK profile looks the way it does.
Yeah, I think another potential big use of this kind of product is to pre-screen animals as well, looking for pre-existing antibodies. I know there's a lot of talk about reducing the amount of animals and potentially reusing them on studies and things. So if you you can screen your animals to know there's a pre-existing antibody against your drug product, then this would actually one, help reduce the animal use, but also it also help when you actually start dosing the animal.
Yeah, exactly. And the final advantage here is that rapid screening is needed before you have done the assay development, and then there's no need for specific assay development for every project. Only those that are going into the clinic then would require that level of validated assay development.
So this gives an overview of the general generic ADA approach. So you have your circulating ADA, which is bound to the drug in the sample. We know that the drug in general has very long half-life, so you get the drug complex kind of being there for quite a long time through the profile of the PK. And then in terms of detection, your antibody is captured, antibody drug is captured using anti-human IgG, and then you detect using an anti-species to detect for example, the monkey antibody that binds to the human drug. The advantage of this is there's no dissociation from the ADA, so you don't need to do an acid dissociation procedure, and also you don't need to label your drug. If you're doing a traditional bridging format, then you need to label your drug. So for example, with biotin and Alexa. In this case, you can set up the drug without having to do any reagent preparation.
Yeah, I think this represents a real advantage from having to do the traditional labeling approach. And in terms of sensitivity, I know that the labeling of the drugs can have a big impact on that. And it's one of the things that we have to really optimize as part of a traditional bridging assay approach.
Yeah, I think the other advantage of Gyrolab is that the microfluidic flow through, and also then the column as part of the microfluidics has quite a high binding capacity. This means that we're going to pull down pretty much all of the drug, onto the column. So it means that drug tolerance isn't an issue. We're not trying to fight with the drug concentrations to measure the free ADA. And it means that we can potentially measure ADAs right at the start of the dosing, or when an immune response appears, particularly when traditional free ADA assays would not detect them.
Yeah, maybe at a high level of dose early on in a repeated dosing scheme is right when you'd start to see that immune response starting to peak out, and so it's a good place to start looking. So let's take a look at what's contained in the kit reagents for our generic ADA assay. Here you can see we have a detection of an immune complex, really, rather than targeting the unbound or free ADA. So this is an anti-species IgG detection reagent coupled with an anti-human IgG capture reagent.
Yeah, I mean, as has already been stated, we don't need assay development, so you can use generic assay conditions, and it really does help get an assay up and running and operational very quickly. The actual reagents will bind a full IgG, but also will bind Fab and Fab2 fragments. So this gives you a lot more flexibility depending on what your molecule that you're developing. Here we show some representative assay performance. So we have a positive control that can be supplied, which is a monkey-human fusion protein, and this can be used to look at the assay sensitivity and kind of the analytical range. And here we've used this to generate positive controls, which we show good performance in terms of precision and accuracy.
Yeah, and this certainly behaves better than a typical assay, but gives you some way to sort of do a QC check on your how the assay performance is working.
Exactly.
So here we have some representative data that shows how the assay performs on a bioaffy 1000 nanoliter CD. And what you can see here is that the acceptance criteria are well within what you would need for a typical ADA assay. And then beyond running the assay in the 1000 nanoliter CD, we have data that shows it could be done. We get similar results if we use a bioaffy 200 or a bioaffy 4000. So depending on the range that you're looking to cover.
So this really shows you the workflow for the generic ADA assay. So you pipette your samples into the PCR plate. You load the plate CD buffers onto the Gyrolab platform, then you run the method using the 1000 method that is indicated here, and then you walk away, and it's around an hour and a half later you'll get the results, and then you review the data. So it's very quick and allows you to run the assay very quickly and get the data quickly.
Yeah, and I think, compared to the typical 4 to 5 hours from a typical ELISA assay, again, you're saving quite a lot of time using the Gyros method.
Yeah.
So what we've tried to do today is to really highlight how the Gyrolab generic ADA kit can fit into your workflow. It's really a ready-to-use solution that offers some convenience and expedited ADA screening for your preclinical work. So again, no need to label reagents, no real assay development, and one ready-to-use kit for any IgG-based therapeutic is what is on offer from the Gyrolab solution.
And it's obviously fast. You can get results in just over an hour, and obviously reduces variability, being fully automated. You can still use the Gyrolab to develop drug-specific ADA assays, so that's another option, but this gives you a lot of flexibility. You have an assay that they can use quickly and is off the shelf, and I think that's really something we wanted to provide people the option to do
too. And I think the last point reflects sort of the understanding that we recognize that labeling the drugs and the labeling process can be onerous. And so we do have custom assay services available that can help out with the labeling process as well if somebody has a specific need in that area.
Yeah, and also what that can do is reduce some batch-to-batch variability when it comes to labeling reagents as well, which can be important, particularly for long-term use of ADA assays. Just as a general reminder, we do have Gyrolab ADA protocols. These are drug-specific assays that have been developed and for various biosimilars, and can help you set up drug-specific assays very quickly when you are, particularly when you look into measure things in human matrix.
Yeah, and I think these are relatively useful for folks that are, even if your drug of choice isn't among these listed in the protocol, this gives you a framework for how you could approach a specific ADA assay on the Gyrolab platform. Well, John, we've come to the end of another Gyrolab Spin video podcast.
It's been from Rob, as usual. What we want people out there to do is send us suggestions. We need areas that people want us to cover. We're happy to, record further podcasts, and with the aim of releasing at least one podcast every month.
Yep, and if you're interested, you can hit the subscribe button on our YouTube page. That'll ensure that you get notification whenever a new podcast has been released. And also, for those out there that are watching, really appreciate your attention, and reach out to us if you have any particular technical questions or needs. John and I are happy to get on the phone or answer emails, and we'd love to interact with folks out there. So with that, I'll say goodbye from me.
And a goodbye from him.
And until next time.